So, hello everybody. We are going to have a look on the questions first. So there are 3 sets of questions for Group A preconceptional exposure. So it's essentially for epidemiological studies in humans, experimental models, and mechanistic approaches. So just to check if people have identified some questions that they would like to be discussed in the group, or if there are other comments or additional questions yes, additional questions if there are some important aspects that we haven't mentioned. Terminology, sure, yeah.
Generation or intergeneration.
Yeah.
Would you like to try to reduce temperature a little bit?
No.
There is no possible.
If there is any air conditioning, but there isn't.
So you want to try? No.
Yeah, so
is it some control or not?
I asked about it and they said that this is all testing.
Oh.
Okay.
Yeah, it's a bit warm.
We get the heating on.
Actually, so I don't want to be provocative. Outside is very nice. Nice weather, very nice. Yes, it is. So if you don't need our presentations, if you go out, just have a discussion.
It's lighting and great. It's lighting and video, I think.
Yeah.
And this was recorded. So we have a free culture, free people.
Okay, so for the epidemiological studies, there's nothing to add. Of course, we can discuss the endpoints, but that was raised a bit earlier. The repetitive sequences and that kind of things. What about the next one, the experimental models? Can you put the next slide, next page on the...
Shall we discuss something about the question that Dominic always raised about the doubling dose for mutation rates? If we have any evidences to, not the equation itself, but the data we have that would allow to support or revise the existing concept.
Yes.
What do we have, what is missing, what we need in addition to be able to revise or keep the same formula from that probably. We also should touch well in discussions.
Doubling dose. We can do that. It's a difficult question, so I think it will...
That I volunteered to answer.
He mentioned that this is mouse data,
Yes.
mouse results from the 90s.
Yeah.
Okay, so we all know what technologies that were in the 90s to get mutation.
Right, yeah.
Okay, so now
Specific loci and...
and
Yeah, it's about really the question 11, transferability of results from animal experiments to humans. That's very much
related to the doubling dose.
doubling dose, yes.
It's doubling dose for mutations calculated, measured from the
Right.
the 90s using all technologies and some low sizes.
Mm-hmm.
Do we have the same doubling dose using next generation sequences.
Okay.
I think it's possible to find results.
Okay. Maybe is it related to in the next slide? There is a mechanistic approach. There is the question 18. Can we link it in that? With what mathematical models would be useful, and how might they be best integrated?
That's actually a question even before the one you were going to say.
Yeah.
Is the one that Dominic proposed, is that the right model, or should we be looking at something else?
Yes, okay. Well, we'll see how far we get. We have tomorrow still.
Maybe on slide 8, which endpoints are the most pertinent? So we are discussing mutations, but is it any mutations or mutations associated with Is it? I'm not heard here. Or? It's for the online. Yes, I was saying that in terms of endpoints, we heard today discussions about mutations, mutation rates, but nothing was said about association of those mutations, association of those aberrations with specific cancer risks. For example, leukemia is known not to be associated with mini satellite instability. So those results from Dubrova studies. So at least leukemia may not be associated. And also the question about the normalization. Are we talking about the same things? Do we want to capture detriment or any change? Like transgenerational effects, traits, is it a change in traits or detriment? So how important it is to actually capture detriment, risk of detriment. So these are the points of the discussion.
It's a detriment, any change, I mean, maybe some changes which currently don't look like detriment may become detriment. There is the change. I mean, I would say any change.
I say maybe, but do you know that? Because maybe
no.
If we have data for these specific mutations, maybe next step is to look for literature other results associating with the disease. If associating, great. So it's good. If not associating, so we label it, okay, this is for further investigation, so for further monitoring. We're not dropping it as umbrella, we just have to go all the way down to the disease problem.
If you look to group D, you're asking what's the relevance for policy, then it would be detriment. You don't want to have a radiation protection policy for something that's neutral.
Well, this is the whole purpose of this calculation. It's a detriment calculation. Right?
It was a question, I think, in the next experimental model or mechanistic, where what is the role of whether Mendelian disease or chromosomal changes or so on. And just referring to what you said, whether it's detriment or any change, so we shouldn't think only of traditional genetic diseases, but there are lots of diseases which are multifactorial or polygenic, and things that currently we think it's not a detriment because it cannot be directly linked to a disease. It might contribute to a polygenic disease or to a multifactorial disease. Yes, plus epigenetics, yes, which complicates the picture.
Very much.
That was also then, is there susceptibility or Mendelian? So I think it is any change.
and the perception issue, because there's going to be a lot of unknowns about what that change might contribute to exactly.
So maybe it actually translates converts to the recommendation, hopefully, when somebody designs an animal study to look at the genetic effects, transformation of genetic effects. So maybe there should be some kind of a facilitation recommendation to go all the way to the disease. Since it's an animal study, anyways, we have these mice, you sacrifice them at some age, maybe you just wait for 6 more months and measure some disease. And then it's a complete picture. You have genetic mutation burden information, and then it converts to disease or tissue pathology of disease. So it's a more complete information, more complete package, at least in animals.
Are there implications for ethical approvals? Well, I'm just saying that I don't know, I don't like regarding those models.
I'm thinking yes.
If you leave animals to then develop disease, I think there are issues. Like that, no?
There are also the epidemiological cohort of offspring, I mean the Maya blocker cohort offspring, and another tertiary cohort offspring, and the full data analysis on hematological malignancy, mortality, and solid cancer mortality been published, and not much been seen. And also we learned from Eric, that that F1 also didn't show much of the detriment effect among them in terms of malformation of cancer. So that we also could link with the epidemiological studies in humans having at least 3 cohorts where the cancer is being looked at. Yeah.
The previous Melody workshop with OECD was actually on adverse outcome pathways, so not these rights from the initial event pathways. The health outcome, and I think that it should be reflected here as well.
Yes, when we have all this molecular and then all the way to the disease data from a single study, the weight of evidence of such result is much higher for the I hope that's important for the disease, frequency of the disease.
It's a useful formalism to systematically approach this kind of question that we have now. Should we look at a change? Yes, if we know there is a link, or if it's plausible there is a link. Yes.
Going to interrupt, one of the members online actually commented that she cannot hear much.
Okay. So
you
might get support is good.
Thank you.
I don't know if it works. We have to work. We have to use it when we speak, each of us. Otherwise, it's a bit difficult. So we were discussing the adverse outcome pathway approach that is now applied quite a lot in radiation research, radiation biology. And there was the previous Melody workshop last year, which was with the OECD to discuss this approach, and it was concluded over this table discussion that this would be a good model for the genetic transgenerational effects as well. Okay, so wonder if we could now move to my presentation and we can discuss based on that. So don't hesitate to interrupt during the presentation. I tried to collect some information really for the basis of discussion rather than a very thorough presentation of the state of the science. So these are addressing, just briefly describing the fundamental biology of male and female gametogenesis. We already heard from earlier presentations today something related to this, and then we can compare if we arrived in the same conclusions or not, or if I spotted those aspects. Next one. Oh, yes, of course I can operate it, sorry. So these questions are more about the experimental models and study design. So I didn't go to the epidemiology, it's more about the germ cells and gonads and the different phases, and thinking about the sensitivity. And there is something also about the species, which are, and their sensitivities. I don't know if I can say what are the best models for humans, but they do have different sensitivities at least. And how many generations should be followed? That's perhaps we should discuss separately. All these, I think the three last questions, I don't have much in the presentation. Okay, so perhaps we go to the next. Sorry, short memory. So now this lovely terminology. So I already spotted the intergenerational inheritance, and it should be added there. But are these definitions okay? And there was the comment about the traits. My point here is that we usually quite often use the terminology genetic effects to point not only the diseases in the offspring, but also biological markers. And we may use this terminology in cell-to-cell progression as well, and not necessarily on hereditary effects. And then, so this for the experimental models, I think this is quite important to have understanding what we are talking about. So, of course, in this group we are discussing the preconceptional exposure of germ cells, so that's before fertilization, and heredity or inheritance of biological inheritance is the passing of traits from parents to offspring. In comparison to the genetic effects, I mean, or the markers, passing of markers. So markers are something that we can measure, but then again, it has impact via the traits, and traits can be also related to this disease outcome. But of course, these definitions are more general, and it's not directly related to the radiation risk assessment, but scientific.
Yeah, I mean, in the morning, when I was listening to all these things, I was thinking about whether I question now that whether hereditary is the correct word to use, right? Because, Mueller and Mendel, they used this terminology before DNA was discovered, right? So the context were different than now, because when we say that breast cancer is hereditary, for example, has anyone studied, the children, second generation children for breast cancer susceptibility? I don't think so, right? Only from the parents to the children. Yeah. And genetic models of BRCA1 and 2, lethal, embryonic lethal, and they don't survive. If a child has, or if the after conception, if you have BRCA1 deficiency, then I don't think the embryo will survive. At least in mice, it wouldn't survive, right? So then it can't be hereditary into the second generation, right?
Well, they. Yeah, well, my understanding is you wouldn't say that cancer is a hereditary disease. I agree. It's a susceptibility, and I would not. Are you sure about that? The BRCA mutations are not
In the mouse models, it is lethal. BRCA1 mutation is
lethal.
Yeah.
Okay.
So they always make what is it called? Organ-specific mutations.
Right.
BRCA1 and 2 are. Yeah.
Yes, I was going to say that in human population, it's heterozygous mutations, BRCA1, BRCA2, that predispose to breast cancer. So I think if you cannot have homozygous, it's also embryonic lethal in both mouse and human.
Yeah, the question is whether heterozygous mutations will be transmitted.
Right.
Because
they follow the Mendelian.
No, that's during Mendel's time. I know, but just to be, we can debate on that, right? Andres here.
No. Well, yeah, but I think I don't have a problem with hereditary effects, because even though comes from Mendel, but the problem is, I think, with epigenetic effects, which are not hereditary. So maybe we should indeed use transgenerational effects, because they are much broad, or this term is much more broad. But yeah.
In the calculation of detriment, I learned this morning from the lecture of Enara Clero. I understood that it is calculated for F1. It's not calculated for F2, F3. So maybe that clears the question about how many generations. One of the questions. We were even for F1, we have, like, our scientific information is so limited. Even for F1, we have so many questions. So maybe we should not consider for F2, especially given for radiation protection purposes, we don't need F2. We need F1. Okay, so maybe that's one question cleared, I hope. And actually that relates to the transgenerational, intergenerational, multi-generational. So F1, it is not transgenerational. So maybe hereditary is a better term.
No, because preconceptional can be transgenerational. Yeah, so it's in utero, it's not considered as.
Preconceptional, sorry, yes, you're right. Yes, I agree. Okay. And also, from the lecture this morning, I learned that the contribution of the hereditary or transgenerational into the total detriment, health detriment, is quite low. It's 0.2 out of 5 at a given dose. So it doesn't.
Taking into account F1 only.
F1, yes.
F1, that would mean that it covers dominant effects and effects linked to X chromosome. Not recessive mutations. I think so. That's what happens. So it's really more about the question whether you should take into account the recessive mutations or not, because for those you have to take into account several generations to get both alleles.
But still, my point is the contribution is low compared to the rest of the health detriment. And should we take that into account when we consider, for example, very unknown epigenetic mechanisms? Should we focus on what we are more certain? For example, genetic alterations. We discussed genetic mutations. It's easier to measure. It's already in the formula, mathematical formula, that Dominic showed today. And I think it's enough to do for the next several years in our studies, epidemiological mouse studies, with a focus on the radiation protection application, like genetics and disease. And epigenetics, maybe we should say we don't know much. There's so many different things happening with epigenetics. There are so many different epigenetic modifications, profiles. We monitor it. We put it on a monitoring level. When we have a chance when we have epigeneticists interested to join these studies, to do these studies, of course it's all good, but maybe there is no such a great need to decide on some epigenetic modifications and how to account for them. I'm just like as a scientist, we don't know. We don't know about epigenetic alterations and their relevance to health. And now we're adding the irradiation component, confounding factors, etc. So.
Yeah, but that's what makes it so interesting. But I just, because Dmitry you were stressing the problem of detriment, and yes, of course, this is relevant. But I think that at least from the perspective of Melody, we are interested in basic mechanisms, and then the application comes later. So while I think that detriment, sure, we have to keep this in mind, I would say, or maybe in parallel, we should not lose out of sight the fact that we just want to know. So in my opinion, it would be very interesting to look at generation 1, 2, 3, because we just don't know. And we know from the Dutch study on the Second World War hunger study that epigenetic effects last at least 2 generations, so maybe after radiation as well. So let's have a look. And then what consequences it has, this we will see later.
I agree, actually, and I also think that it's part of another question, but being able to sample and think about the range of different markers, I think that's when we need to be getting as much out of each sample as possible. I definitely agree with going the multiple generations and F2 and F3 for all studies for the reasons you've just said.
Yeah, I think that the way it has been previously done, so the 2007, it says 2 generations, I guess. It was calculating for the 2 next generations. And before that, it was calculated over a longer period. So, but I think we have to check this maybe by tomorrow. And if we want to take into account or give the possibility that epigenetics is taken into account, then it's, by definition, you have to go to F3. Okay, then about the terminal, yeah.
There was a preprint by Braun Schumacher, C. elegans transgeneration histone linker study. I think anyone has read it. Just last month, he just put up the preprint. Yeah. C. elegans multigeneration.
Yeah, I'm sure there are in different species, but the C. elegans also has quite interesting reproduction, I think, yeah, which is very different from.
But relatively speaking, those comparisons are very complicated.
Yeah, sure.
Lower organisms need very high dose.
That's true. That's true. The dose comparisons, but they do have, of course, the advantage of short lifespan, and you can study several generations, but very difficult to draw conclusions to humans. That's the problem, and doses and that kind of things. Ike, you had.
So for the generational rational thing, could we agree that from a scientific point, it probably makes sense not to set a limit whatsoever because we want to understand, but then when it comes to recommendations for policies, we could also say that we currently see no indication for taking into account more than one or maybe two, just because for the lack of scientific evidence. So it would be very interesting to study it in more detail and hopefully get more information. But right now, there is no such indication and not even enough to say, well, we should be prudent, so let's look further into the future. Yeah.
I work with the
research needs.
Professor Nomura. I was in his lab, and I don't think he has published all the studies he had done. He has done multiple generations, and we used to go to the animal house in the morning and spend whole day by just observing them. And the last publication he had was in 2006. I don't think he has put up any experimental data in that publication. Actually, he has spent a lot of money and thousands of mice for multiple generations.
Well, F1 is to decipher the mechanisms, and as a biologist and scientist, this is, well, maybe the most interesting, but thinking at population level, multiple generations would give an insight of the degree of deterioration of populations or changes in population fitness. I mean, this is less studied in humans, but if you think of various animals or other organisms, this happens, and why it happens in humans also, just we don't know.
And I guess another reason you highlighted on it with the cohorts, the F1s are mainly very aged, and same with the study that we've been doing, and the F0s, so the F1s are 50, average age 50s, and so actually to be able to follow through, just from thinking about the confounders and all of the things that make everything noisy and difficult. So that's.
Just add something about, because I'm not sure whether when you were talking about recommendations for detriment and so on, are you talking now about from perspective of the workshop or from perspective of the task group? The difference is in that the task groups do not give any recommendations. The task groups summarize science. Yes. Yeah, that is what Chris always tells us also in TG121. I mean, there are some members, this is the individual radio sensitivity. We want to give recommendations. No. You just put together the science. That's it. So just to warn you, not to go too far, because it's going to be a dead end.
It's difficult to set the border, but it's very small. I think it's important to understand what kind of knowledge, scientific knowledge, summarized scientific knowledge, is taken by the advisory groups, and then first UNSCEAR, and then ICRP, and then regulators. How this knowledge converts to regulation,
right?
Yeah, but that's outside of the task group. Yeah, it's a different.
But then how do you determine priorities, research priorities? What is not known?
We have strategic research and that's in the platform. This is outside of this debate.
Okay, so you want me to stop?
No, we can discuss this, but it's not to the topic. This is not, we are discussing about
okay, let me put it this way then. Let's discuss scientific knowledge linking genetic mutations or genetic mutational spectra or epigenetic mutational spectra to disease, which is relevant to the task group. So
yeah, when when we were writing up the Unscare report, I was summarizing all gene expression data from radiation exposure, and none of the reports mention about the disease association. You know, it's impossible for us to associate genes from others' publication. I think it's very, very difficult. Even the authors themselves, they don't link gene expression changes into any disease outcome in humans.
Yeah, because the question is not the question that most people ask that do gene expression studies in terms of their responsiveness to radiation exposure is the shape of the dose response. Yeah. And here again, I would like to come back to what you said about trying to figure out which changes are related to which disease. Yes, it is interesting, but again, it may be outside of the scope, because the shape of the dose response, is there a threshold? Is there no threshold? And that is really important. And also, the question on which effects to focus, which genetic effects or epigenetic effects to focus on, which one gives detriment and which are maybe neutral. Yeah, that is interesting again. But on the other hand, we all suffer from lack of statistical power in studies. So if you start to say, okay, no, I will just look at this particular effect, probably you can draw no conclusions. So this is also something that we must bear in mind, that it's the same. I mean, if you work with mouse strains, you select a mouse strain that is susceptible to a particular effect in order to potentiate the effect. Otherwise, you're going to spend millions of dollars or euros looking at very rare events.
Well, actually, I put on this slide which is a bit in the same topic. Endpoints, what endpoints are evaluated and what is the health outcome expected. Just taking up some: cell death, mutagenesis, epigenetic changes. And what would be the outcome? Well, from cell death, you would expect that it affects fertility. Mutagenesis, that would be hereditary effects. And then you have to think about the selection, removal of severe damage, and how the chromosomal aberrations are linked with reduced fertility as well, and cell death. And epigenetic changes, okay, that would be the transgenerational effects on gene expression. So we don't know what it means in terms of hereditary effects. There's much more information on epigenetic modifications that are seen in somatic cells and their relation to cancer, for example, or other diseases, but we don't really have much data on the germline. And then there are different steps during the sperm maturation in fertilized egg, and that when much of the epigenetic marks are removed, the reprogramming that was discussed earlier, so how much and what kind of markers will prevail and what effect might they have. So it's quite a big task to find out all that. Anybody willing to comment on this aspect? So, but anyway, I think it's very important to try to discuss the endpoints in context of the health outcome expected.
Yeah, let me comment. Of course, it is important. I fully agree with you, even if we don't come to a conclusion. But just to lay out what do we know, what we don't know. But I think that if you want to see which changes in the genome or epigenome are associated with which consequences, you have to look outside of the radiation research field. You have to look into the medical field, and that becomes a huge task. At the same time, we have to remember that the majority, I mean, in humans, we don't have a single trait that is dominated by a single gene. It's all multifactorial. It becomes very complicated. So even if you have, I mean, we see this when we try to correlate in GWAS studies, to find an association between a certain map or scenario of SNPs and the disease, and you don't find this. Or you find extremely weak links because it's so complicated. So again, maybe we should. I mean, it's fine. We should discuss and lay out things, but at the same time, remember to be modest because it's very complicated, and I don't want to discourage anybody, but be careful in your aims. I think in the aims, because we are entering a field that is extremely complicated.
Yeah, I think if we think about cancer biology or the susceptibility loci for cardiovascular disease, they usually have hundreds that are associated. And then it's about the combination. And so that it is very, very complicated, and it's impossible actually to have just one gene, except for the ataxia telangiectasia and that kind of, yeah. But they are rare cases, they are not for the public health. Yeah. Yes, please.
So then we have, wouldn't it make sense to say that we have a substantial number of human cohorts, F1, maybe F2 also, and in some of these cohorts, genetic mutations are measured. The rate of genetic mutations is measured, like Chernobyl, Japanese cohorts, some others. So maybe we should say that it is most appropriate to continue and complete studies in those human cohorts and measure epigenetic changes, do more comprehensive genetic mutational profiling, and then also these cohorts will be followed for detriment, for disease still, because this is our primary interest, and say we focus on this. So this is our primary source of information for understanding the health risks. In terms of mechanisms, so Andrzej, you said you're interested in mechanisms. Melody is interested in mechanisms, not in regulatory issues. But I believe mechanisms is understanding how mutations, which mutations, epigenetic modifications, how they actually contribute to pathology. And this is also AOP. I know you're skeptical about AOP in general. No, it's okay. Sometimes I'm very skeptical about that as well. But if you say mechanisms is the primary interest, then I think you cannot just limit yourself to studying those responses from mutations and epigenetic alterations. You need to understand how these mutations are, which mutations can be associated with the risk of disease. And an example is chromosomal aberrations. There's many studies looking at the chromosomal aberrations because we know that people with increased levels of chromosomal aberrations in blood lymphocytes, they're predisposed to cancer, etc., etc. So I think it is, we cannot ignore completely information. Yes, we need to gauge our interest and the amount of effort. Like you said, we cannot do mouse studies that take 10 years and millions of euros. But I think we still, if we're interested in mechanisms, we still should pay attention to some of the associations with disease when we do molecular studies.
Yeah, I partly agree with what you said just at the very end, that, following human cohorts raise a lot of questions in terms whether we will really understand the mechanisms due to the very low incidence of the outcomes and so on. So I would say that in order to answer these questions or these highlighted endpoints, we should think of finding a suitable experimental model for the purpose of studying the mechanisms. And this, in my opinion, these are only animal models, because in human even in other epidemiological cohorts, cancer incidence and so on, even there we have problems with the low incidence and so on. Now here with the transgenerational effects and or hereditary effects and exposed individuals, the expected outcome is even low. So I would doubt that you can find cohorts big enough to be able to study really mechanisms. You might be able to validate or to test mechanisms found in animal experiments. So I think a key question here is to think of what is the best model to study, really to study the mechanisms.
Yeah, I think the points raised earlier, I didn't get his name. Dimitri, yeah. So most of the cohort studies are post-exposure effects. You can't probably, it's very difficult to associate changes happen before, right? For example, when I was analyzing Mayak workers' chromosome, I was suggesting that we should retrospectively go back and check chromosome alterations and the cancer outcome in that particular population, but I left Colombia that time, and then they did even send me the slides, but those slides were the glasses were so thick, I can't even fit into my microscope, because they were prepared in the 1960s or 50s, 60s and 70s. If those studies would have compared, then we would have had a perfect model for chromosome alterations and disease, because they have the data from cancer incidents in that particular cohort, and big number, right? But unfortunately, there is no prospective study.
Yes, and commenting on the epigenetic changes, when we look at the susceptibility loci, for example, for cardiovascular disease or cancer, many of these loci are in the non-coding regions. And that's an indication that they impact gene expression rather than, they are not mutations in specific genes, but in between. And it's regulatory areas. Yeah. And it's, well, if we think about the human cohorts, there are some limitations, like it's a sensitive issue and ethical questions, quite a number of them. So, and we are indeed the lifespan is very long for humans, and we cannot go very much beyond the F1 anyway.
Yeah, exactly.
At the moment.
Only mouse studies with genetic,
but
retardation mice, for example.
Yeah. Okay.
If the mutation is detrimental, there is no way you can have progenies.
Depends how that.
No, the removal from, severe damage will be removed from the population. Then there is no way it can be transmissible, right? Very difficult. I think you have the meiosis slide there.
Sure. That's one option. What may happen?
Exactly.
But the changes, these potential hereditary changes or mutations, they can be very different. Nonsense mutations that don't have any impact.
Yeah.
They can be even beneficial, and then some of them are bad.
But they can be detrimental.
Yes. Yeah, exactly. Yeah. What could be, well, I don't know directly what could be an example of point mutation that is lethal. It's mainly perhaps the chromosomal alterations. Yeah.
So there was some mention of massive deletions in the
yeah,
morning.
but again, it's chromosomal level
Yeah.
already. It's big.
Yeah.
Okay. So I tried to look at the DNA damage and repair in human reproductive cells, and let's see what can we extract from there. Yeah, there's the point on the spermatogenesis. So DNA repair is efficient in spermatogonia and spermatocytes, but deficient in spermatids and sperm. So again, that's when you are exposed, it's quite critical if it's in this respect. If it's the sperm which is exposed, there could be the higher chance to for the mutations. And then there is the fact for the reprogramming. So there are protamines are replacing about 80% or perhaps more of histones during the sperm maturation. So this is something protecting from environmental stresses the sperm, but on the other hand, all these a very large proportion of the histone modifications are removed. So there is the question how much is really transferred to the next generations. And in the oogenesis, that's really on the borderline if it goes to the group B, but anyway, of course the sensitive stages are during the embryonic development. Oocytes are very old cells. So they are formed in the embryonic stage, and they are arrested in the first meiotic division until ovulation, and then meiosis is completed upon fertilization. And after the fertilization, there is another reprogramming. There is a reprogramming step for epigenetic marks and reprogramming. In addition to the reprogramming, there is also repair of sperm damage done by the oocyte. So that's an interesting point as well, thinking about it. More or less in meiotic cells or germ cells, there are the same repair mechanisms that are available for in somatic cells. So in that sense, it's not anything special, so to say. And the oocyte's ability to repair different types of DNA damage, there is a repair machinery that can act on sperm DNA damage. And there are even some numbers given in the literature, so that if oocyte has the capacity to repair sperm DNA damage when the level of sperm DNA damage is less than 8%, which is quite high. But then higher levels of sperm DNA damage are associated with a failure to reach the plastocyst phase, and the embryo are not viable. And again, of course, the single strand breaks and abasic sites, they are more easy to repair than double strand breaks, but the mechanisms are there, and But anyway, a mutation means that the DNA damage has been repaired. So it has been repaired, but it's wrongly repaired, so there's a mistake. And some points about the oocyte sensitivity to radiation. There are age effects. So developing gonads inside embryos are more sensitive because there are rapidly dividing cells that are more vulnerable. And then there is some indication in general that germ cells from older individuals may be more susceptible for non-disjunction and mutagenesis. There are species differences on the sensitivity, and okay, the strain differences that were discussed, they have been observed in bred or outbred strains of mice. The stage of growth of the follicle, which surrounds the oocyte, has an impact, and the primordial follicles there are considerable species differences. So I found this information that they are highly sensitive in mice, rats and rabbits, but then again, humans, guinea pigs, monkeys are highly resistant. So perhaps it has something to do with the repair and the histology of the ovary as well, and oxygen levels and that kind of things that were discussed earlier today. And I don't know who has these multi-layered follicles, which species, but they seem to be more variable. But there are species differences in the sensitivity of the oocytes and ovarian.
Can I ask a question in this connection?
I don't know if I can answer.
Because we talk about radiosensitivity, and these cells are more radiosensitive than the others. But what are we talking about? Killing? So do we know actually that if a cell population is sensitive in terms of killing, that the radiation effect, a radiation exposure would lead to fewer mutations in the survivors? Or is there a correlation between a high level of killing and a high level of induction of mutations? I don't know.
Yeah, but it could be that. Yeah, well, I don't know.
I don't know. This is a good question.
If it's not fragile, then you may have a lot of killing and a lot of mutations as well.
In the survivors.
Or perhaps it's if there are differences between the individual cells, those that are resistant survive.
Yeah, yeah, but maybe
I don't know.
Yeah, I suggested
also have a question or comment. So with all this information, we can probably model a situation when we know the time of. Let's imagine it's an acute exposure of a particular case scenario. So we know the exposure time of parents, and we know what at which stage the germ cells were for the exposure. So we know that, for example, the child will have higher risk or lower risk, but apparently for radiation protection, it's not like we're not supposed to use radiation protection for individual risk assessment, right? It's all for radiation protection to regulate. So question is, how useful all this information, like basic biological information about the sensitivity of various stages of germ cells biology for the radiation protection system? Because we cannot use it for, not directly, yes.
Informative, can we transfer any species from the human? Because the question is species that would be similar to humans.
And in a population, you would have all the different stages at the same time. So if you understand the relative contribution of each, that also gives you information about the mix as a whole. And that's pretty much what's done in radiation protection.
Yeah, see, when you go for diagnostic radiation, right, they'll take your undertaking that you know that it's damaging. Similarly, probably when people sign up to go to space travel, so they should have signed informed consent that I know the risk, right? That information scientists can give to the public. It's not policy, it's not radiological protection, but the basic understanding of radiation.
So if we think about the scientific knowledge, we have epidemiological information, we have experimental animals information from that, some understanding on the mechanisms and the function of human genome and cells and so on. And then if we are lacking information in epidemiological side, how do we fill in this gap?
Again, I'm not the lab person, but for me, it's clearly that some of the animal models could be useful for the understanding of mechanisms and risks in humans, but that's clearly also the difference between the spermatozoids and oocytes, where, I mean, for me, it's really interesting, and I have a question about any data on the transgeneration effects in guinea pigs. Monkeys, I don't know, probably it's very expensive study to do and follow generations, but are there any data on that topic from guinea pigs? It's mainly mice and rats and, no?
I don't know. In fact, this information was from the 1970s, so when they did a lot of animal experiments, so it's not recent information, it's there.
It would be relevant for us to look at this information?
I think so, yes, go back and yes, with new eyes, so to say.
This one can be potential. One potential caveat is that in those years, in those times, they used a lot of high-dose experiments. And whether high-dose experiments are convertible to low-dose experiments is a question, because we had a discussion just a few minutes ago that killing of cells, which is more likely at higher doses, might lead to some uncertainties, whether it's actually clearing the population from mutated cells or inducing more. And this is less likely situation with lower doses, because lower doses are less likely to induce cell killing, more likely to induce mutations. So I think we have to be careful about translating high dose experiments to this.
It's not that I always contradict what you're saying, but please don't take this. We are discussing. But because what yes, it's an interesting point. But on the other hand, if there was an overkill effect at high doses, we would see a saturating dose response for stochastic effects, and we don't. We either see a linear or linear quadratic. So I'm not sure if this is really a problem. The fact
in mice you do see
saturating,
for example, for leukemiogenesis and cancer.
Well, but at very high doses. But I mean, sure. Well, you have wait. You have these bell-shaped effects. Yes, of course. Yeah, but that is beyond what I think what people have used in terms of doses in the 1950s, because these are the experiments that you refer to, 50s and 60s, when they were doing wild animal experiments. Really. Yeah. Okay. Are we? Okay. Yeah. Okay. Okay. See. Okay. Good. I take it back. It depends on the dose.
Might be relevant for therapy, maybe. Okay. So then some consideration about the epigenetic signal and what are the criteria. So for the somatic cells, it would be a mechanism capable of transferring signal or mark to daughter cells. So it's from one cell generation to the next ones. And then you have to prove that the signal or mark is there in the daughter cells, and that signal or mark should have impact on gene expression. So what it would mean then for germ cells and transgenerational effects is that you have this, there is a mechanism capable of transferring the signal or epigenetic mark to next generation, and then prove that it is there. You can measure it in the offspring, and then show that it has effect. I don't know if this is helpful for any. But if you really want to go from the mechanisms to health outcome, how can you do that?
Do you mean for the somatic cells? You mean it's a more in vitro system, in vitro experiment? Because it's a daughter cell, it's a cell division in a petri dish. Dividing population. Okay, I think it's a very good question point. How much we can trust in vitro experiments in somatic cells with epigenetic or genetic endpoints, and convert these results to organism scenarios? Because it's like, to me, it's a big question. I'm not an epigeneticist. I don't know, probably some knowledge can be transferred, but I think it should be, like, we should be careful how we convert in vitro results to in vivo organism. Because you clearly say daughter cells, and then you say generation, next generation, which to me is an organism. So I think we have to be careful.
Yeah. Sorry, I'm going to have to stand up. My back is gone. Apologise, I can't stretch. The additional thing at the end, what we talked about earlier, going from gene expression to actual health or some change in pathology, would that be an additional point to put at the end?
Yeah, this is epigenetic signal in general. So the gene expression, if we think about the detriment, yes, it should have some impact.
The line over there where you're relating gene expression change to some sort of pathology change.
Right, okay.
Be linked to some sort of
so that from risk to be informative for risk assessment, there should be some detrimental outcome. Okay. This is more or less to just to comment on, people see some changes in gene expression. They are, as such, not informative for epigenetics, so it has to be transferable to be epigenetic. Some discussion about the transgenerational epigenetic inheritance. Again, thinking about the situation that exposure of parent has impact on characteristics of the non-exposed or exposed offspring. So that's more or less the definition. And if we think about risk assessment, it should be then the characteristic should be something that has detrimental health outcome. And it is known that various environmental exposures may cause these changes in the acquired state, like they say, which is the characteristic. And acquired states of gene expression and characteristics that may alter the epigenome of germ cells. And then we discussed already a bit this information transference to the next generation. And the point is again made here on the DNA methylation, which is reprogrammed in the germ cells of the embryo or the zygote, actually. And there will be another reprogramming after fertilization in early embryo. So how much information is lost? This reprogramming is actually very interesting from the biological point of view as well. The babies do not accumulate all the mutations and damage that is there in the parent generations, but they are renewed.
We will be extinct by now.
So there has to be some selection. It could be selection, it could be this reprogramming, it could be DNA repair that is renewing the developing individual. If you don't get rid of the burden, so to say, it could lead into death of the early embryo.
Can it also be random? I know very little what epigenetics.
Well, why not? I don't know. I have no idea.
In terms of
Random. So yes, there are random mutations as well, you know.
So they would effectively have a half-life of so many generations.
Yeah. There are these, if you think about mutagenesis, there are replication errors that are kind of background, and it's not caused by environmental agents. But errors occur, and I see no reason why it would not be true here for the epigenetics as well. It looks very random anyway, you know. There's a lot of different kind of changes, but we just our knowledge is not enough at the moment to judge.
Maybe we should. I was thinking also. Do we really have the experimental means to study, especially this last point you say? So the changes of epigenetic patterns over time in multiple generations in vertebrates, mice, and well, humans and so on. But maybe we could sort of take help of plants and lower animals, yes, fish and whatever, to find a pattern, because I'm not an expert by far in this field, but I'm not aware of really experimental means of studying these kind of longitudinal changes in the epigenetic patterns.
We have group C.
Mm-hmm. Yeah.
It's a mouse model to see the effects of genomic DNA methylation and disease that are associated with obesity. So this mouse model can be used, but I agree with the other animal models. For example, zebrafish. I was just discussing yesterday with a colleague how zebrafish is actually has been useful for human cancer, understanding human cancer mechanisms and finding actually drugs to treat cancer. So because 70% of the genes are overlapping between zebrafish and human, and if you do lead studies on zebrafish to identify candidates, and then do validation studies in the mouse, I think that's a powerful combination. And last thing, neurodevelopmental effects in zebrafish are very much relevant, like the pathways of neurodevelopment, very much relevant to human, mammalian and human. And we can use potential neurological consequences in F1, F2 in zebrafish. So yes, zebrafish is a relevant model.
What is the low dose for the zebrafish?
Which media? Water,
Yeah.
air, biological tissue.
Yeah, about this reversibility, I think there is information that they do disappear within a few generations, so they are reversible, and epigenetics is even used for cancer therapy because you can reverse the signaling and yeah.
He mentioned this.
Yeah.
But he also had one case that we went on for a long time.
Epigenetics, not only that it is reversible, but it's also highly affected by environmental factors. So, reversible or changeable.
Perhaps. Yeah, perhaps there could be in this case even recommendations when you have been exposed. You can reverse something. Healthy lifestyle, which is always recommended,
No, I don't know whether you can say that it is reversible because we know about dominant mutations, right? If the breeding is made between animals of similar mutations, then you can say that it is reversible. If one parent having a mutation, the other one is not having mutation, then you have dominant effect. No, there will be resetting during fertilization. Reprogramming or resetting. So whether epigenetic mutations in one and the other one, which one will take dominance, right? See, the thing why I'm telling this, I worked on telomeres for a long time, and one of our collaborators did work on two mouse species, one bulb C and Muspratus. You have in bulb C 40 kilobases of telomeric DNA, Muspratus has 15. When you cross, the dominant one was, you expect that there will be an average telomere length. So the Spratus telomeres are completely gone. You have only the longer ones remain. Maybe similar mechanisms, because whatever markers or epigenetic parameters you need will survive, right? Will take over the weaker ones. I mean, that's just the concept is. No, there were studies done where the resetting of telomeres occurs after fertilization. Otherwise, continuously you lose telomeres just like telomerase knockout mice, but that doesn't happen. When people measure and associate telomere length with any disease, I would ask the question that what is the functional relevance, right? If you continue start losing telomeres, you will be extinct. There should be resetting during meiosis, or there is resetting because it's proved. That's what a similar thing may happen in epigenetic markers, who knows?
Yeah, about the schedule, I just noticed, it's almost 6 o'clock, and we have Vadim's presentation tomorrow, maybe. Tomorrow. Yes. Or do you wish to have it tomorrow as well?
Tomorrow also she can speak.
Yeah. Okay.
She can continue tomorrow.
That's right.
Actually, I see this contribution is very limited here, and tomorrow I will speak probably. But if you allow me, I can.
Okay. Yeah.
To bring more optimism into the generally vague situation.
So write this, I'll make tomorrow. Yes.
I was just participating in the final discussion this morning about Singapore grant review panel. They want to look at telomeres and dietary patterns in mothers.
So there is my name. Something with Irfan. 6 melody, yes, correct.
Yeah, this one.
Okay, so actually, so my participation in today's session was quite inspiring for me, because before I thought that the most vague part of epidemiological studies is dosimetry. Now I see that not. Dosimetry is the most precise and definite, and maybe sometimes robust part of the studies. So today, Zhenya made overview of trio studies, and she mentioned this one study, And so I will tell you about the symmetry. No, don't ask me about endpoints and about genetics. It's not my area. It's very complicated for me. But in general, so there is a study which lasted for 5 years. And the idea was to see whether de novo mutations are associated with exposure. And as you heard today, there are design is to have to score both parents and offspring, and see whether there are something new in their genome, in the genome of offsprings. So altogether, doses were reconstructed to almost to 300 trios, although only 105 were included into analysis, which went into the paper in Science, which did not find any mutations. Speaking about doses, so we had to reconstruct gonadal doses, of course, for females and males, different target organs, but they're located almost in the same region, so it was not much difference for dose calculation. And what is important, we covered doses from the moment of Chernobyl or beginning of exposure till two dates. One is date of birth minus 38 weeks, which is called here DOB 38, or another till 51 weeks. And the reason is that there are different moments, different critical moments were considered, and we had to reconstruct two kinds of doses, which were included into the analysis. Also, the end of the period, if the date of birth was later in time. So for cleanup activities, we stopped dose calculation in 31st December of 1990, which is technically the legal limit of cleanup. After that, it was regular work without any special emergency or accidental connotations. But for other components, which I will show you a bit later, we calculated till the DOB 51 and DOB 38. So we had to consider different pathways or components of doses, and for the dominant, and you will see later, the main contribution was from cleanup activities, from cleanup mission, external exposure. Also, in our study, we had 5 different groups. Again, I will explain it a bit later. And for some of these groups, like wives, which are not liquidators themselves, but they were in Chernobyl, we calculated external radiation doses while they were in Pripyat before evacuation. And also, we consider doses in other settlements than Pripyat. That means that virtually all territories of Ukraine are contaminated, and there is some extra Chernobyl component, and so we estimated this part of exposure as well. So those reconstructions was blind. That's why don't ask me about any other aspects of the study, because actually, so we were called to reconstruct doses unbiased, without any relation to outcomes, to effects, etc. However, we knew, of course, to which of the groups belonged, 5 groups belong a subject or trio. So we used analytical or another more historical classifications, time and motion methods, which is Radru-Rockville. So they originally were developed for a series of leukemia studies, which included Ukrainian American in the cohort of Ukrainian liquidators, and another study which was led by IARC, Elizabeth Cardis at that time, which covered cohorts of Baltic state and Belarusian and Russian liquidators. And also we had to modify this methodology a little bit. So the idea is to make interview of a person, ask about his places and time and what he or she did during the cleanup activities. And then the results of this personal interview are read and interpreted and translated to certain formalism of this Druer-Rockwell methodology, the software and input interface. And then doses are calculated, including stochastic analysis, which means 10,000 realizations for each individual dose. So for this study, we had to update a little bit this code to make more detailed dose reconstruction for time spent in Pripyat, because in the original version of Rockwell and Druer, it was simply like any other places, but in Pripyat it's urban environment with buildings, with shielding, with different floors had different shielding, different dose rates. So this module was significantly revised and improved. And also for external dose reconstruction, the respective conversion coefficients, like from conversion from air karma, those in open air, to gonads were applied. So now we are coming to the study groups. So there are five different groups, and first four are combination: father exposed, mother unexposed, mother exposed, father unexposed. Both were exposed, both are not exposed. And interesting was category E. We picked some fathers with high doses, which led to acute radiation syndrome. So the subject with acute radiation syndrome.
So I'm going to ask, why were there no mothers with highly exposed persons? There were no such cases.
Virtually, yes. Among those who with acute radiation syndrome, there are only a couple of women who were not personnel of the nuclear power plant, but they were kind of guards. But as far as I know, they died. So there are no alive high acute radiation syndrome female subjects. So that's the point. But in general, in the population of liquidators, women are only 5%, and in this high dose, virtually zero, because they were personnel of firefighters or personnel of the nuclear power plant, which are all males. So coming to the doses. So some of these numbers were already presented by Zhenya, but I prepared this slide before, and it's with different inclination, from different point of view.
Based on the larger number of subjects, because what was published.
Not only, but also the way how it is presented. So first of all, we reconstructed these gonadal doses, and you can see that for fathers, all groups, doses are much higher. And the upper bound is 4 gray, one of those subjects with acute radiation syndrome. Mothers usually received much lower doses. However, there was one case with pretty high dose. If we consider those contribution of different components I mentioned, the highest was due to cleanup activities for liquidators as liquidators. The next was for residents of Pripyat, and you can see that so female are red and male are blue, like color identification of babies. So I don't know how it is in Europe, but in Ukraine, so if boy is born, all things are gifted in blue, and if girl in rosy. Okay, so that's why I make this remark. But okay, so I marked the traditional way. So you can see that, for instance, for Pripyat, since the ladies spent all the time while men were partially in work, the dose is a little bit higher for them. And also that it's really small doses, but not completely negligible doses from other Chernobyl sources, non cleanup, non-Pripyat doses. In most of cases, doses were these 2 different doses, which make a lot of troubles for dosimetries to calculate separate procession. But in most cases, they are identical. This within 1 milligrade, simply because of stochastic modeling. So we did 2 different rounds of stochastic modeling, and as a result, doses are slightly different. But for 35 fathers, it's different because of conception time. So here is a summary of all data, and you see this all groups. What is interesting is that, for instance, group D, non-exposed, you see that we discovered that some of subjects were actually exposed. So it's about the presumptions. So we made presumption. Originally, we picked up those persons as not being exposed to any significant Chernobyl radiation, but indeed we had some. All other are quite predictable. However, for instance, for group C, both parents are exposed. You see that mothers still received lower doses than fathers. So here is a summary table. The most important, I suppose, is the number of trios in each category. So we try to make that it's even, but almost succeeded, not completely. But you also can see that the number of children is larger than number of trios, because some of them had duplets, twins, and or not necessarily twins, but duplets, and 4 had triplets. Well, here are the arithmetic means, median, and range. Actually, for epidemiological analysis, it's a long story, a long fight between dosimetrists and epidemiologists. Why use arithmetic mean? Because those distributions, particularly the stochastic dose distributions, are very strongly skewed to the left. So they're like log normal, and GSD is quite big. So the more stable and more representative metric is median dose, but still epidemiologists prefer arithmetic. And you can see that they are much higher than the median doses. However, in case of, so there are some attempts to make stochastic epidemiological analysis, and in this case it doesn't matter. They simply use this 10,000 realizations, and Mark Little is doing that. So I think it's also it's my final point in my presentation. Okay, so that's it.
And what was evident actually is the small number of people you need for epidemiological study compared to. Yeah, but still, it's feasible in that sense. Mutation analysis takes time, but.
It's true. You said at the beginning that there was 300 or more individuals interviewed, but they've not been studied. Are they going to be analysed in the future, or what was the reason for not selecting them?
So it was a very short presentation. I didn't go too much in details. So the original idea was to pick up those subjects from existing cohorts. One cohort is a so-called clinical epidemiological cohort, which is run by our research center for radiation medicine, and those people are followed up for medical examinations. Idealistically, every 2 years, but not real. But still, there is a contact with those people, so they were picked from this. This group E with highest doses is another sub-registry for acute radiation syndrome. And I'm not sure about the numbers, but some were picked up from the National Chernobyl Registry. But it was random selection. And originally, when the study was conceived, the target number was 450 trios, father, mother, and at least one child. So we ended up with almost 300, because it's really difficult to enlist people, to trace them, and so that's what we had. And our epidemiology and genetics colleagues were impatient, so they made analysis based on 105 trios. Did I answer?
I guess I still want to know if the 105 were, for instance, in your groups that are clinically followed up, and also will there be some sort of an association done with any clinical effects?
So I don't think so, because I think it was kind of it also a blind study in this sense. So it was not associated with any apparent medical problems. So it was simply taken saliva samples, and they were, I think it was full genome sequencing, and then they look at certain loci, which, don't ask me which what, it's not my area.
Sorry, yeah, if you know the mutation to
link, because there is a cancer registry, not for other diseases, but it's possible to link the study subjects, children, with the measures in the population cancer registry to see if any of them cancel it or.
Yeah. That's right, and so the contact with those people is existed. So it's if necessary, it's possible to. I heard this discussion about whether we will look at simply some mutations, or mutations associated with some detriment. Yeah. So in this study, it was not a purpose to look at it.
Yeah, I mean, so the study that I'll describe tomorrow morning, it's a very similar study, much smaller, and by no means is there any dissymmetry associated with it. It's military veterans of historical nuclear testing. So we have, we're not in a position to be able to do this dissymmetry that you've done, but from very limited information of self-reporting information, then it would be useful to be able to do some sort of analysis or evaluation across the different endpoints. I mean, like your study, everything is done blind, but actually when everything's decoded, it provides an opportunity to do it. And I'll hopefully explain that tomorrow.
So yes, it's possible to do in the future, no question. But also according to the protocol, for instance, it was decided not to provide feedback to subjects. But several reasons. One, unless there is something very threatening for them, so then it's not directly communicated, but put to the consideration of IRB, whether we did not have such. And one of the reasons was that, I don't know the number, but I am sure that there were some. Like in any such kind of study, about 10% of children have different others than expected. So then it's one of the reasons why not to.
That's a common problem.
Yes, but it's a matter of the personal situations. We don't tell. Yes.
So the bottom line is it can be done in the future.
In principle, yes. So if there is some interest, so write me or I will communicate to our opponents.
Also ask a question about the uncertainty in the dose estimate, because it's based on interviews. Did you validate it somehow, or how? I mean, or do you have something about the uncertainty?
Okay, so in principle, the methodology was valid. Well, tomorrow or on Thursday, I invite to my oral presentation, and I will touch this issue. So this methodology was very thoroughly tested, validated, and against different data, like gold standard was EPR dosimetry with teeth. Another is there are some subset of reliable individual monitoring data, not many, but also for high doses we compared with dicentrics and some fish also. So it was validated very thoroughly, and in our calculations we made stochastic modeling, which accounts for part of the uncertainty. And this part of uncertainty, which we call an intrinsic, which related, for instance, to uncertainty in the dose rate data, historical dose rate data in the dose rate maps. I did not have time to explain much about the methodology, but the idea is to overlap the variables collected in a course of personal interview with the dose rate maps, which are pre-calculated, and so this accounts for this kind of uncertainty. And now, this uncertainty is on average about GSD about 2. That's to give precise answer to your question. Sometimes it is as high as 2.7. So for dosimetrists who are doing this dosimetric monitoring is too much. So typical uncertainty is 1.3, 1.4. tolerable too, but it's exceptions. For epidemiologists, they're happy with that. So that's my answer. But there is also another part of uncertainty which we call human factor uncertainty. For instance, some people can invent some heroic events, some forget something which is very important. So we made some small sub-study on that, and we published it recently, and I can send you a paper if you want. So it might be the same order of magnitude. So there are two kinds of uncertainties combined. It might be larger, but still it is quantifiable. It is quantified. It's communicated to epidemiologists. That's the best we can do.
Sure, no, I understand it. It's interesting to know the dimension.
So what is your first reaction? Is it too much or too low?
What's the best that you can do? I mean, what else can you do?
Yeah.
I mean, this is what we are interested in finding, of course, biomarkers of exposures. And then if you add those, yes, but we don't have it.
Pardon.
The online participants.
Oh dear, yes. Yeah. Time is up, more or less,
I'm happy to pick this up tomorrow morning because I've just had a really long day. There's a few things that I think that would be there's obviously an overlap in how we've been trying to do things, but we've got a much larger cohort and a lot more information.
What's are humans? So it's combinable.
Okay, thank you very much for these discussions today, and let's continue tomorrow.